Carbohydrates are essential compounds made up of carbon, hydrogen, and oxygen. They are absorbed as monosaccharides or disaccharides and play a crucial role in our diet. Carbohydrates are classified into sugars, polyols, oligosaccharides, and polysaccharides, each with specific components and functions. Today, we will explore isomerism in carbohydrates, including various types such as stereoisomerism, optical isomerism, and mutarotation. Stereoisomers have the same molecular formula and atomic connections but differ in their three-dimensional shapes, requiring bond breaking to interchange. Polarimetry measures the optical activity of chiral molecules, which do not rotate polarized light and racemic mixtures. Von 't Hof's rule helps determine the number of isomers based on the number of chiral centers in a molecule. Epimers are sugars that differ only in the configuration around one carbon atom, often seen in glucose and galactose. Enomerism occurs in sugars with the same molecular formula but different arrangements around the anomeric carbon atom, forming alpha and beta anomers. Mutarotation in glucose involves changes in optical rotation due to the equilibrium between alpha and beta anomers. Simple sugars have common names and can be classified as aldoses or ketoses, depicted using Fischer projections. D-glucose is an aldose with 4 stereogenic centers, commonly found in various foods and used in the production of syrups. The ring formation of D-glucose involves the creation of alpha and beta anomers through cyclization. D-fructose, the sweetest sugar, forms a ketose structure and is a major component of honey and fruit juices. Ribose, a pentose sugar, is a key component of riboflavin, nucleotides, and nucleic acids, with a unique structure in DNA. D-galactose, found in lactose or milk sugar, forms a ring structure similar to glucose but with different configurations. Hexoses can form various pyranose structures, each with unique properties and functions. Disaccharides are formed through condensation reactions, joining 2 monosaccharides with a glycosidic bond. Isomers in disaccharides, such as sucrose, maltose, and lactose, have different glycosidic linkages and structures. Lactose, a disaccharide, is formed by the linkage of glucose and galactose, showcasing the diversity of glycosidic bonds. Monosaccharides and disaccharides are fundamental carbohydrates, each with unique structures and roles in our diet. Sugar alcohols, also known as polyols, are a group of organic compounds derived from sugars. They include D-mannitol, D-sorbitol, dulcitol, D+arabitol, adonitol, xylitol, and L-threadol. These compounds are commonly used as sweeteners and in various food products. Polysaccharides are complex carbohydrates formed by the combination of multiple monosaccharide molecules. They can be classified into oligosaccharides and true polysaccharides based on the number of monosaccharide units. Examples include starch, cellulose, and glycogen. This table compares cellulose, starch, and glycogen based on their source, subunit, bonds, branches, diagram, and shape. Cellulose is derived from plants, starch from plants, and glycogen from animals. Each has unique structural characteristics and functions. Starch is a heterogeneous material composed of amylose and amylopectin. It forms helical coil secondary structures and is a significant component of plant storage. Starch granules can be found in nanoparticles, which have unique properties due to their size. Amylose and amylopectin are 2 components of starch. Amylose is a linear polymer, while amylopectin is highly branched. Both have distinct structures and properties, contributing to the overall functionality of starch in plants. Glycogen is the primary form of carbohydrate storage in animals, particularly in the liver and muscles. It is highly branched, allowing for rapid release of glucose when needed. Glycogen's structure is optimized for efficient energy storage and retrieval. Cellulose is the most abundant organic molecule on Earth, forming the structural component of plant cell walls. Its unique linkage and fiber-like structure provides strength and rigidity to plants. Glycogen, starch, and cellulose have distinct structures and branching patterns. Glycogen is highly branched, starch is moderately branched, and cellulose is linear, reflecting their different roles in biological systems. Understanding isomerism is crucial for predicting organic reactions, proposing mechanisms, and developing safer and more effective drugs. Isomerism plays a significant role in the specificity and selectivity of chemical transformations. Test your knowledge by identifying the monosaccharides in lactose, maltose, and sucrose. This exercise helps reinforce the understanding of carbohydrate structures and their components.